Welcome back to the revision series of human physiology. Today we will do quick revision of breathing and gaseous exchange. So first, some basic facts about respiratory organs in non-vertebrates. So simple organisms like sponges, coelentrates and flatworms. You can remember this from your animal kingdom chapter also. Sponges, coelentrate, flatworms, etc. They can simply diffuse different air molecules through their skin. Earthworm, it has moist cuticle. It can simply diffuse oxygen and carbon dioxide through its skin. Insects have a tracheal network. You will learn about this tracheal network in structural organizations in animal cockroach part. Next, there is aquatic organisms which obviously have gills. So aquatic arthropods and mollusks, they obviously have gills. Plus, in vertebrates, aquatic vertebrates like fishes, etc., they also have gills. And then there are terrestrial non-vertebrates, etc., which also use lungs. Next, moving to vertebrates. In vertebrates, fishes use gills, obviously, whereas reptiles, birds, and mammals respire through lungs. Now, one important thing that you need to remember is the amphibians like frog can respire through their moist skin also. Now, as you may remember, in non-vertebrates, earthworm, and in vertebrates, amphibians, they respire through their moist skin. And this form of respiration is called cutaneous respiration. Mammals have also developed a respiratory system which involves, as you know, lungs and all the air pathways part. Moving to the important part of human respiration, this diagram you must already be familiar with. So here is the nasal cochlea and nasal cavity. Moving down, we have larynx, which is the voice box. Then we have trachea, which is separated by esophagus through a flap-like cartilaginous structure called the epiglottis. And this trachea then divides into two branches which go to left and right lung respectively. You already know about the structure of left and right lung, and then it further divides into bronchus, then bronchioles, and then into alveoli. We will look into that. So as you know, the trachea divides into right and left primary bronchi and the level of fifth thoracic vertebrae. This trachea branches out into right and left bronchi at the level of fifth thoracic vertebrae. Next, each bronchi undergoes repeated division to form secondary and tertiary bronchi and bronchioles, ending up in a very thin terminal bronchioles. They asked this several times. So, after trachea, all the other primary, secondary, tertiary bronchi and bronchioles, they are supported by incomplete cartilaginous rings. So from trachea to the initial bronchioles, all the structures are supported by incomplete cartilaginous rings except terminal bronchioles. Next, terminal bronchioles give rise to a number of thin, irregularly walled, vascularized bag-like structure called alveoli. Now a double-layered pleura is covering the lungs. In between this pleura, there is pleural fluid present. What this pleural fluid does is it reduces friction when the lungs expand. So when the lungs are expanding, the pleural fluid present between the two pleura will reduce friction. Now conducting part transport atmospheric air to alveoli. As you already know, it clears it from foreign particles and humidifies it and brings the air to body temperature. These are the four functions of the conducting part. You also need to remember that thoracic chamber is anatomically airtight. Now another very simple part of this chapter is mechanism of breathing. So as inspiration happens, lungs expand, so volume increases. Now since volume is increasing, the pressure inside the lungs is decreasing. So because of the negative pressure, lungs have less air pressure in them and the atmosphere has more. So air from atmosphere goes into the lungs, and as expiration happens, it happens because of positive pressure. So the volume of the thoracic cavity decreases while expiration, and this causes lungs to have more pressure and atmosphere to have less pressure. So the air goes from lungs to outside. As you can see, air entering the lungs and air being expelled from the lungs. Now you need to remember that while inspiration, diaphragm is contracted So the diaphragm contracts during inspiration, and while expiration, diaphragm is relaxed and arched upwards. Abdominal muscles also help in forceful expiration. So when abdominal muscles contract, forceful respiration occurs. On an average, a healthy human breathes 12 to 16 times a minute. It's a general fact, you already know this. Spirometer helps in clinical assessment of pulmonary functions. This is also important. Next, lung capacity. These are things that you have to mug up. There are 4 basic lung volumes, and after that, mixing up this lungs volume, you will get different lung capacities. So first there is tidal volume, which is 500 ml. Inspiratory reserve volume, which is 2500 ml to 3000 ml. Tidal volume is the generally air expired and inspired without any forceful expiration or inspiration. So inspiratory reserve volume is how much air a person can take in more just after a normal inspiration. Expiration reserve volume is how much air a person can expire after a normal expiration. So after a normal expiration, which will be the tidal volume, you can still remove some air from your lung, and that will be called expiratory reserve volume, which is 1000 ml to 1100 ml. And then reserve volume is 1100 ml to 1200 ml. This is always present in the lungs, and you cannot remove this air. This is present so that the lungs don't collapse on themselves. Now, inspiratory capacity is the total volume of air a person can inspire after a normal expiration. So after a normal expiration, you can inspire tidal volume plus IRV, because that much space will be present in your lungs. You can do the math and find out that how much is the inspiration capacity in human, which is tidal volume plus IRV. Expiration, total volume of air a person can expire after a normal inspiration. So you can do a normal inspiration, which will be tidal volume inside your lung, and now expiratory capacity will be that tidal volume plus the reserve volume which is there in your lung, not the residual volume, the reserve expiratory volume that is there in the lung, which will be ERV. So TV plus ERV. Next, functional residual capacity. This is the volume of air that will remain in the lung after a normal expiration. So you can see that functional residual capacity. So this will be expired. So this is the inspiratory reserve volume. So this tidal volume will be expired, and this will be the expiratory reserve volume plus the residual volume. This will be present in your lungs after a normal expiration, which will be tidal volume. Next, there is vital capacity, which is the maximum volume of air a person can breathe in after a forced expiration. So you can see that after a forced expiration, this is gone, and this was not there. So only the residual volume is present in lung after a forced expiration. So after a forced expiration, first of all, you can take in the tidal volume, then you can take in the inspiratory reserve volume, and then you can take in the expiratory reserve volume. So you can take all of these 3 reserve volumes inside your lungs after an forceful expiration. Moving to the next important part, there is exchange of gases. So exchange of gases between blood and tissue occurs through simple diffusion, which is based on pressure concentration gradient and solubility of gases, as well as thickness of the membrane involved. We'll move to the thickness part later. More the solubility, more the exchange of gases will be enabled, and less the thickness, more the exchange of gases will be enabled. Therefore, the wall that is present between the alveolar cavity in an alveoli and the blood vessels that cover the alveolar cavity is very thin. So there is only 3 layers of cells that are present. One is the basement substance, which is present between the alveoli and the blood vessel, and then there is the alveolar wall, which is 1 cell thick. Next, there is the endothelium of the blood vessel, which is also 1 cell thick. So it is a very thick wall which is present between alveoli and blood vessel. So this is the partial pressure of oxygen and carbon dioxide at different part in the respiratory pathway. as compared to their partial pressure in the atmosphere. So as you can see, atmospheric air has very high partial pressure of oxygen 159, and atmospheric air has obviously less partial pressure of CO2 0.3. Now this gets exchanged in deoxygenated blood, because in deoxygenated blood CO2 is more, so CO2 is 45, PCO2 is 45, and PO2 is 40. And obviously, as you know, in oxygenated blood PO2 will be higher, so PO2 is 95 and PCO2 is 40. Now in tissue obviously has more partial pressure of carbon dioxide, because a tissue is using oxygen, so it will have less oxygen, so PO2 will be less, 40, and PCO2 will be more, 45. One more important fact that is given in NCERT and that you need to remember is that solubility of CO2 is 20 to 25 times higher than that of O2. Transport of gases, in this part there is few very important things that you need to remember. All the facts, few very important facts are crammed into this one paragraph. So first you need to know that about 97% of the oxygen is transported by RBC in the blood, as compared to 20 to 25% of CO2 which is transported by RBC. So 97% of O2 RBC and only 20 to 25% of CO2 RBC. Next, the 3% of oxygen is carried in dissolved state through plasma, as compared to the 7% of CO2 which is carried through the dissolved state through plasma. So as you remember, so again 3% of O2 plasma, 7% plasma. Next, what about the remaining 70% of CO2? The remaining 70% of CO2 is carried as bicarbonate in the RBC. We will look into this mechanism later. Moving to oxygen, as you know, oxygen is carried as oxyhemoglobin. Now this is another important fact. Each hemoglobin molecule can carry a maximum of 4 molecules of O2. Yes, as you can see in alveoli, where there is high PO2 and low PCO2 and lesser H positive concentration, that means less acidic and lower temperature. All these factors are favorable for formation of oxyhemoglobin. We will look into the graphical form of presentation of this information later. First, you need to remember these 4 factors which are favorable for formation of oxyhemoglobin. Next, these are the 4 factors that are not favorable for formation of oxyhemoglobin and are favorable for dissociation of oxygen from oxyhemoglobin. So dissociation of oxygen from oxyhemoglobin will obviously happen in the tissue. So the 4 factors responsible for dissociation of oxygen from oxyhemoglobin in the tissue are low PO2, high PCO2, high acidity, and high temperature. Next, you need to remember that 100 ml of oxygenated blood carries 5 ml of O2. You already know that each hemoglobin molecule can carry 4 molecules of O2. So 100 ml of oxygenated blood can carry 5 ml of O2. This is the oxygen dissociation curve, which is the graphical representation of just what we studied up. So in this, the y-axis represents the percentage saturation of oxyhemoglobin, and the x-axis represents the partial pressure of oxygen, which is in the units mmHg. So as you know, this is the partial pressure. This is the curve which exists, and it is a sigmoid curve. So the oxygen dissociation curve is a sigmoid curve. As the oxyhemoglobin saturation increases, partial pressure of oxygen also increases. So as the partial pressure of oxygen increases, the oxyhemoglobin saturation also increases, but in this manner. Now, when oxygen dissociation curve shifts towards right, this indicates a reduced affinity. We will look at the practical implication of this in the next photo, but first you need to know that as the oxygen dissociation curve moves right, it reduces affinity. It happens because of, as you know, all those things which causes the dismissal of oxyhemoglobin into oxygen and hemoglobin. That is increased temperature, increased acidity, and difference in PO2 and PCO2 concentration. Obviously, the left shift shows an increased affinity towards formation of oxyhemoglobin, and these are also because of just the factors that we studied above, which aids in the formation of oxyhemoglobin. Now this is the practical application. As you know, this is actually hemoglobin. Sigmoid oxygen dissociation curve is actually a characteristic of hemoglobin. But when we talk about myoglobin, which is another compound which is present in muscle. Now myoglobin does not have a sigmoid oxygen dissociation curve. Myoglobin has oxygen dissociation curve in this manner. You can see this light blue myoglobin, which means its affinity towards oxygen is obviously high because it is present in muscle. So it has this sort of curve. And as you know, this left shift is caused because of high oxygen affinity. And also one important point, this is the fetal hemoglobin, the dot line. Now fetal hemoglobin, it is also present in the left of normal hemoglobin. The curve is present in the left of normal hemoglobin. That means it has higher affinity than the normal hemoglobin. But the fetal hemoglobin, it is present in right of myoglobin. The curve is on the right of myoglobin. That means it has less affinity than myoglobin. This is how you can practically use the oxygen dissociation curve in finding out which chemical has more affinity or less affinity towards forming compounds with oxygen. Moving to next carbon part. So carbon is transported as carbaminohemoglobin. Now as you know, 70% of carbon, as we have studied above, 70% of carbon is transported as bicarbonate. So what this means is, in alveoli we need CO2, which is inspired and expired through lungs. And in tissues we have too much CO2. So what it does is, as PCO2 in tissue is high, the PCO2 gets converted into HCO3 negative. Because PCO2 is high, so CO2 gets converted into HCO3 negative, and which joins H positive and travels in blood in the form of bicarbonate. Now this conversion of HCO3 negative to H2CO3 occurs due to carbonic anhydrase, which is present in the RBC. This enzyme is present in the RBC, and this H2CO3 moves through blood to the alveoli, where the PCO2 is less in alveoli. So since the PCO2 is less, H2CO3 gets converted into CO2 in alveoli and H2O, since now CO2 is less. So now since we have CO2 in alveoli, this CO2 can be expired and inspired. Now this CO2 joins H2O again in the alveoli with the help of the enzyme carbonic anhydrase, which is present in the blood vessels near the alveoli, and this gets converted into H2CO3, travels to blood, and the same thing happens again. So it gets converted into HCO3 negative and H positive. Simple thing that you can remember is that in tissue, the form in which this CO2 is present is HCO3 negative. In tissue, the form in which the CO2 that is transported as bicarbonate, 70% of CO2 is present, is in the form of HCO3 negative, since the PCO2 is high. And in alveoli, since the PCO2 is low, the form in which carbon dioxide present is obviously CO2. Next, every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to alveoli. Now, every 100 ml of oxygenated blood, as you remember, delivers 5 ml of CO2. Next, now there are only two more important parts that are left in this chapter. First is the neural and other centers responsible for regulation of respiration. First is the pneumotaxic center, which is pons, which can reduce the duration of inspiration. It does not do anything to expiration, but it can reduce the duration of inspiration, and thereby it can alter the respiratory rate. Next, there is respiratory rhythm center, which is present in medulla. Now pneumotaxic center, which is pons, can actually moderate the functions of respiratory rhythm center, which is medulla. And now a chemosensitive area is also present adjacent to the respiratory rhythm center, which is highly sensitive to CO2 and hydrogen ions. And you can read this whole part which is there in NCERT. And increase in these substances can cause this center to signal to respiratory rhythm center. There are certain changes that need to be made in the respiratory process so that these substances, which is like H positive and CO2, can be removed. So chemosensitive area can indirectly signal the respiratory rhythm center which is present in medulla. Also, the pneumotaxic center can also moderate the function through respiratory rhythm center. And what does basically pneumotaxic center do? It reduces the duration of inspiration. Now, these receptors, there are certain receptors which are present outside the brain, which are present in the aortic and carotid artery. So these can also identify CO2 and H positive concentration and send necessary signal to rhythm center for the remedial action. So in this you must have observed two things actually in this whole paragraph which is there in NCERT also. First, respiratory rhythm center in medulla is the main center which coordinates breathing. So in the medulla oblongata, respiratory rhythm center is the main center which coordinates breathing. So for example, pneumotaxic center also does its function through the respiratory rhythm center. It moderates the respiratory rhythm center. That means it sends signals to respiratory rhythm center and tells it what to do. There is a chemosensitive areas that are present near the respiratory rhythm center that also send the signals to respiratory rhythm center. Next, there are the receptors which are present outside the brain, which are present in aortic and carotid artery, which also send their signals to the respiratory rhythm center. And the second thing you must have noticed is that these receptors, the chemosensitive area and these receptors, they can recognize CO2 and H positive concentration, but not O2 concentration. So actually the role of oxygen in regulation of respiratory rhythm is therefore quite insignificant, which is a line given in NCERT also. Next, moving to respiratory disorder, there are multiple respiratory disorders related in breathing in this topic, but in NCERT there are only three important given. And since we will be covering, since this is revision series, I will only cover those three disorders which are given in NCERT. First is asthma, which is difficulty in breathing due to inflammation of bronchi and bronchioles. So in asthma you need to remember that there is inflammation of bronchi and bronchioles involved. Next, there is emphysema, which is a chronic disorder actually, in which alveolar walls are damaged. Now, as obviously you can make out that since alveolar walls are damaged, the respiratory surface is decreased. You must already know that cigarette smoking etc. causes alveolar walls to get damaged. Hence, this causes emphysema. So this is next is occupational respiratory disorders. There are certain industries like mining, in which the miners or the people who are involved in those industry are exposed to certain chemicals for a long period of time, and this causes different kinds of problems in the lungs. For example, this can cause inflation of the alveoli in lungs, which causes fibrosis and certain other disorders. So fibrosis is the proliferation of fibrous tissue present in the lung because of inflammation. Certain chemical can, like asbestos, can cause asbestosis. So exposure to these harmful chemicals during mining and stone breaking and grinding can cause damage in the lungs. Damages or inflammation or fibrosis caused because of these chemicals come under the section of occupational respiratory disorders. This completes the revision of our chapter breathing, exchange and gases.